This is episode 93 of the Immunology Podcast, Cancer Immunoregulation with Dr. Rahul Roychoudhuri. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoyed the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Rahul Roychoudhuri from the University of Cambridge on the podcast to talk about his research on cell therapy and immunity to cancer metastasis. We also got our usual roundup of recent highlights in the immunology news coming up. But first,
how can we forget? We want to remind our listeners about the upcoming Immunology 2025 meeting that's happening on May 3 to 7 in Honolulu, Hawaii. Late-breaking abstract submissions opens on January 7 and closes on January 21st. Learn more at immunology2025.aai.org.
All right. Well, you're going on vacation for a while.
I am. Sorry, not sorry.
I don't understand how you even get 3 weeks of vacation in a year. What is this?
This is the European lifestyle. You should try it sometimes.
Don't you get more vacation in the summer too?
Well, I did not take an extended vacation in the summer because I was saving my time to go now. And as you know, as fellow immigrants will agree, it's not a vacation when you are going home to see your family, really. So it's more of a family obligation.
That's the only vacations you're allowed to get as Americans. You have to use all your vacation time to see family.
Yeah. So by the time this episode airs, I'm going to be enjoying, I don't know, a glass of Malbec somewhere in Argentina.
I have a LARP in late November before Thanksgiving, so I have 2 days of vacation, but I'm flying back from a work trip to go to it.
All right. Okay. You are.
So for my second time in not distant past, I'm packing my LARP stuff with my work stuff. But this is like a more chill LARP. So last time I packed chainmail in my carry-on, and that was interesting.
Well, yeah, make sure to wear the right thing. Don't go with your LARP costume to the business meeting. I don't think they're going to be a little bit confused.
I've done that before, but that was intentional one time.
You need to just, what's the word? Blend in with the suits. You're a suit now, Jason. You're a suit.
Yeah, but I'm a suit who wears Jedi robes.
Still a suit. All right, suit. Why don't you impress me with your favorites today?
Suits, I'm going to talk about colonists.
Okay, sure. Go. Worst. That's the worst segue. You know what? Just go for it.
All right. This is in Cell. It's called Viroid-like Colonists of Human Microbiomes. First author is Ivan N. Zeludev, and last author is Andrew Z. Fire. So this is fascinating. So there are these obelisks, what they call them, because of how they make these secondary structures that are viroid-like RNAs. So they're phylogenetically distinct, viroid-like RNAs that are associated with the microbiome, not the host, and found across the world, more in the oral microbiome than the stool, but in the both. And they form these obelisk-like rods. So they're like these rods can harbor ribozymes. So some type of, RNA enzyme activity. They have an obelin 1 and 2 function. There's a bigger protein who they have a crystal structure they've been able to predict. And then a small 40 amino acid 2 in some of them. And then they found that, in particular, specific bacteria they exist in. But long and short, I have no idea why they went down this path other than clearly they had a reason. They developed a bioinformatic tool called Vinom. Nom, 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 nom. And it's based off of what we know about the hepatitis D virus, which is a similar weird RNA virus that doesn't have a reverse transcriptase in it. So these are similar to hepatitis D in that way. And they did a pan scan to the microbiome from public data sets. in addition from the Human Microbiome Project, and found these suckers. So they found the sequence. They did secondary and tertiary alignments, both the protein and RNA. They're all about 1,100 nucleotides in length. They provide some pictures of this rod-like structure. Public data is replete with these suckers popping up everywhere. And this one bacteria, Staph sanguinis, in the oral microbiome, they use as the model system, and they find it, and they then isolate they get different samples of it, and they find that some bacteria have it, some of the Staph sanguinis have it, and some have lost it. And they can find they find no difference in survival benefit by losing or having the gene just doing bacterial culture, but they find that it makes up a decent percentage, like 0.5% of the RNA transcriptome and a decent amount of the protein as well in bacteria that have it. So they're pumping a lot of it out, and they don't know why. They don't know what it does. It's just there. And they seem to be spread, and you can look at phylogeny and see how they extend throughout the entire world, and they're all related to each other, and. So this is really cool because it's like a whole new, not quite form of, viroid life, air quotes, with these obelisks running around, and no one knows what they do. And it goes in the structure and here and stuff. But like the high level is there's a whole new world out there again.
That's so weird.
I
I'm
love
looking.
it.
I just like looked at the abstract, like just to see if I could understand what you said you meant. That the figures are not very helpful. The one in the graphic abstract. So it's basically just this RNA, because it's not even a virus. Is it like parasitical? Is it like it seems to be like neutral to the host, or does it seem to? It's just there. It's just replicating and just existing inside this bacteria.
No one knows what they do. That's the next step. Protein of unknown function. Da, da, da.
They don't really say how did they come to start identifying. Because
They did. They created a bioinformatic approach to identify them based on known patterns of hepatitis D, which is similar.
Oh, okay. But then how did they know they would go like? How did they learn that thing first, that they were similar to hepatitis D?
Well, so hepatitis D is an RNA virus that doesn't have a transcriptase or anything in it, right? It just is a fully RNA virus that it's bigger, but it's also a bit weird. And so for some reason, and they don't explain why, right? Like they decided to make this Vinod tool what was it? Nom, right? To find it. And then they found a whole bunch in the Human Microbiome Project databases.
Oh, that's so cool.
So why they really went there is like one of those things that clearly they knew something that's not in the paper, but they went on a fishing expedition and found a new lake.
Wow, that's so much fun. Okay. All right. I think that what I'll talk about is also a little bit we went on a little bit of a fishing expedition and found something really cool. The paper I want to talk about is called Endogenous Self-Peptides Guard Immune Privilege of the Central Nervous System. Super cool. First author, Min-Woo Kim. And this is from the lab of Jonathan Kipnis. And at the Washington University in St. Louis. And it was very interesting, very, very nice story. So basically, in this paper, they tried to understand antigen presentation in the central nervous system, and a lot of the Kipnis lab work is around regulation or understanding the immune system in the CNS, and they just the question is, how does it work, this kind of immune privilege, quote unquote, of the central nervous system, the brain. We know that it's not so much like that. We know that there's actually a lot of immune cells in the brain, and you have even like bone marrow, like spitting out immune cells. But there are differences in how the immune responses are carried on in the brain and in other places, in the CNS and other places. And so what they do is they look into kind of they they use immunopeptidomics, and they analyze peptides presented to by MHC class 2. This is in mice, right? In the MHC class 2 in CNS. So they look at the different parts of the brain, draining lymph nodes, and this kind of things. And they kind of look at see which peptides are enriched in the CNS compared to other places, and what do these peptides mean. And it's very interesting because they end up really zeroing down into a few peptides, two peptides that are derived from a protein known as MVP, which stands for myelin basic protein, and which is highly expressed in the CNS. And there's a few specific fragments from this protein, particularly these MVPs 160, 175. They seem to be very highly present and very present in the CNS, in the CNS samples that they make. Interestingly, so they identify this peptides. They see these are peptides that are specifically being presented in the CNS, not in other places that are fairly large, higher levels. So they're clearly part of the normal immunopeptidome of the brain. And what is interesting is that if they immunize mice with these peptides, right? So that's a way, for example, of initiating these MS-like symptoms in mice, is that there's specific peptides that if you inoculate mice with these peptides and an adjuvant, then you end up having a CNS, inflammation and immune response, but not with these peptides. Although they're being presented in the brain, they cannot kind of break tolerance against the peptides, and they cannot generate as a neuroinflammation when the mice are being, inoculated with those peptides. And in fact, what they find is kind of a subset of suppressive T cells. They have this. It's kind of like there are some FOXP3 regulatory T cells, but that's not the group that they are mostly find. They find some CD4 T cells that are not FOXP3 positive, but they do express CTLA4 at large in high amounts, and they also produce TGF beta, which are both of them are known immunosuppressive mediators. And what is even more interesting is that if they inoculated mice with MOG peptide, which does initiate an EAE disease and does induce neuroinflammation, but if they at the same time immunize with this MVP 160, 175, this other peptide, they could actually prevent the inflammation in response to the inoculation of MOG. So not only were these suppressive cells responding to this peptide not kind of inflammatory, but they could actually suppress the differentiation or kind of the establishment of a pro-inflammatory response against a different peptide. So I thought that was so cool. And they look into the thymus, and they do show that this specific cells have a greater frequency in thymus, and they already have kind of a suppressive regulatory-ish phenotype right out of the thymus. Super interesting. And so what happens when you do have institution, when you do have inflammation? What's happening with this cells? Why are these cells not protecting? When mice develop EAE, how is this protection mediating? Why this protection lost? So one of the things is that they show that when they initiate this autoimmune encephalomyelitis model in mice, the presentation of these peptides, of this kind of tolerogenic peptides, is diminished. So if they look at the immunopeptidome, there's less of this peptide being presented, which would presumably mean less suppressive cells are being stimulated. And that's why this tolerance can be broken, and then you have immune inflammation. So really showing that it's an active kind of element that is preventing your inflammation are these suppressor cells, which again are not, most of them are not, what I would call a regulatory T cell. They're FOXP3 negative, but they are expressing a lot of CTLA4, and they're making TGF beta. And I think what's very interesting is that if they deliver this peptide using extracellular vesicles to the CNS, just delivering the peptide can suppress EA by expanding this population of CTLA4 positive suppressor cells. Specifically, including in the draining lymph nodes to the brain. So really this also gives a therapeutic angle to this discovery, and I thought that was super super interesting. So yeah, that's active regulation in the brain.
Super interesting. So again, cells are a spectrum. These binary definitions are so in the past. You don't even need to be a Treg to be suppressive. I guess that's true of MDSCs, right?
You just do what you have to do. There's no label. You don't need to be labeled one thing or another. You just do your job the best way you can.
How do you know what your job is, though?
Well, I don't know. I mean, in this case of these cells, I guess the thymus tells them. And there must be something about that MBP peptide that is activating the cells the right way during thymus development. But yeah, it's very interesting to look at this method in particular. So what you got?
All right. I'm also talking about the brain a little bit, and the heart, and sleep. This is a wild paper in Nature, and I love it so very, very, very much. So it is in Nature, myocardial infarction augments sleep to limit cardiac inflammation and damage. Came out, accepted 23 September 24 in Nature. I believe the first two authors, based on superscript number 21, are Pacific Hune and Jan D. Hoffman, with the last and corresponding author of Cameron S. McAlpine. I'm sorry if I missed other first authors, but I actually can't find where the decoder ring is on the paper here. All right, so this is a crazy story. All right. So after you have a heart attack, people tend to get sleepy. And they found this is actually important for cardiac recovery. And they map out the entire pathway as why. So they do this in mice, but then also look at human data and to validate it. What they do in mice is they give them heart attacks, and they find, using tracing studies, that microglial cells and monocytes, microglial cells activate and monocytes chemotax to the brain. Okay, step one. What are they doing there? They're secreting TNF. That's the next thing they find. They do knockouts and show that this is also important. So you should just assume every step of the way they do lineage tracing and tissue-specific knockouts, because it's a Nature paper to prove every single thing is in here. I'm just trying to robustly summarize. So then in part of this TNF and part of the thalamus, it activates a program in the thalamus, this TNF does, that makes people sleep, that makes the mice sleepy. And if you disrupt, you can then wake up the mice instead, but that makes them sleepy. And by being sleepy, they have decreased inflammation in the heart from immune chemotaxis of immune cells. And that sleepiness has signaling that tells the heart to chill out through decreased activation of adrenergic receptors in the heart immune cells, not the heart cells, and not in catecholamine levels, but in the receptors on immune cells in the heart. There's less catecholamine drive. And so there's less inflammation to the heart in sleepy high TNF brain mice that leads to less post-ischemic damage and better healing. And so they find if you disrupt this by like waking the mice up or knocking out various parts of the pathway, mice have more cardiac rupture, decreased heart function post-MI. If they look at people, do studies of people, they find that people who have better sleep quality have better cardiac outcomes post-MI. And so they tie this all together. But it is shockingly sophisticated. It is a tour de force pathway. So yeah, so this sleep is really important for mediating damage. They get through that pretty quickly. That's only the first half of the paper. Then they're trying to figure out what happens in the immune cells. And so there's immune cells that have this adrenergic receptors on them. So from catecholamine, your adrenaline and norepinephrine and all of that. And so and dopamine, right? And so, but it's really noradrenaline in this case. So they see significantly increased cardiac dopamine beta hydroxylase, where you screw up sleep. If you screw up sleep, you have more of this. And that leads to more localized production, not systemic, but more localized activation. If you knock out the receptor on the immune cells for this, you have less of the effect. So basically, after a heart attack, your brain sends immune cells to your brain, your body sends immune cells to the brain, secrete TNF into the thalamus, which makes you sleepy. And by being sleepy, you have less activation of your of catecholamines in your heart tissue itself, which leads to less activation of that pathway in cardiac and pericardiac immune cells that move there. And that leads to less downstream extra damage. And if you wake people up over and over again, which we do in the ICUs all the time, or mice, then you don't have that decreased catecholamine tone. And then you have worse outcomes.
Dude, that's so far-fetched. It's like super cool.
All the immune cells.
Yeah, seriously. I mean, it really shows that it's good to rest after a traumatic event, I guess. This is a very complicated cascade of events to demonstrate. It must have been.
They use bone marrow chimeras with lineage tracing and then knockouts. Like, let's put these immune cells with this knocked out into this one. They'll take the immune cells, and I don't go into all this because of brevity here, but they take the immune cells that have been toned to, have the receptor knocked out or not, or MI-based immune cells, and put them in other mice and see what they do, or they give heart attack to one mice and immune cells with a knockout into that. So they do all these cross systems to really ferret out like what part is important at what step. And so they'll take one mouse's immune system and shove it in another one, and that mouse may have had the heart attack or not, or the other mouse did or not. And so they can do all these crosses and lineage traces and everything else.
Wow. That's pretty impressive.
It's a tour de force. This is a paper when I read, I'm like, yeah, this goes in Nature. I'm done now.
All right. So then I guess the moral of the story is, if you have a heart attack, make sure to get sleep as crazy after. Don't allow don't wake a patient that is recovering from a heart attack. Let them sleep. All right. Okay. So last story of the day. I mean, this week has been really hard to choose papers because there were so many good papers coming out. So I just had to choose one. I'm sorry for all the other great things that came in the past few weeks, but this paper is also really cool. So this paper is called "Rhythmic IL-17 Production by Gamma Delta T Cells Maintains Adipose De Novo Lipogenesis." And this is also like these crazy connections between different systems. It's just wow. So circadian rhythm, very important. Keeping our body has certain activities in the body change throughout the day in order the night, the day, and that's very important. And we have all these internal clocks keeping our body on this 24-hour cycle. So we eat. The idea is that we do the right things when we are awake. We do the right things where we're sleeping. When we eat, we make fats. When we're sleeping, we can use those fats, things like that. And fat is particularly something that has a very kind of regulation across day and night. And in mice, of course, they have kind of an opposite circadian rhythm to us. They are active in the night. They're eating at night. And so also their lipid metabolism also is different between night and day. When they're eating during the night, is usually lipogenesis is enhanced. And when they're sleeping during the day, lipolysis is more likely. And this is something that people have observed for some time. This is something that's how it is. So what they do in this paper, which the authors, I almost forgot, first author Aaron Douglas from the lab of Lydia Lynch at Princeton University, was published in also Nature, yeah, just plain Nature. Congrats, guys, on another Nature paper. And they study the effect of immune cells in the adipose fat, in the adipose tissue, and how they connect the circadian rhythm and this cyclic fat metabolism. So what they see is that in the fat, there's this innate T cells across tissues, but they focus on the fat. They have very present, this machinery for the clock, for the circadian rhythm, this clock genes, they're highly expressed on this particular cells. And I think basically, and they have it in gamma delta cells and some invariant mite cells as well. But basically, the gamma deltas that make IL-17, they are the highest overall in expressing this gene. And they also the expression of IL-17 by these gamma delta cells is also rhythmic. They see that they see an increase of IL-17 production during the day and reduced during the night in mice, right? That's remember that mice are active during the night, and that is really seems to be modulated by members of the molecular clock. They have some knockout mice and they study this. And if they reverse the light cycle, they can also reverse the IL-17 rhythm and also the metabolism and this lipogenesis versus de novo the lipolysis versus de novo lipogenesis. And kind of long story short, because I don't want to do it too long, what they see is that not only is like do this gamma delta cells, have this cycle, but they're actually important for they influence the lipolysis or lipogenesis, and if you can actually modulate the synthesis or degradation of fatty acids with IL-17 that is made by these cells. Deletion of IL-17 or IL-17A or F, which are different chains, they are impair de novo lipogenesis, and this really shows that these cytokines and these cells through the cytokines are regulating the fat tissue, and that this regulation comes from their own endogenous circadian clock genes that they are expressing in high amounts. And I don't think so. Basically, they show what is someone say? They show that the loss of this gamma delta cells disrupts the de novo lipogenesis, and this is this is measured by reduced gene expression of these genes and at reduced IL-17 levels in the adipose tissues, and basically they see this. I mean, it's very weird, right? You have this fairly small amount of immune cells that are regulating the lipogenesis in the fat. And of course, the question is, what are therapeutic? This gives you therapeutic potential through interventions that are restores circadian IL-17 signaling in metabolic disease. So I guess it's very interesting to see what they could how could this knowledge be applied. But I also find it so weird, so unexpected.
Well, fat is one of those organs we don't call an organ, but it is an organ.
Yeah, that is true. And a very immune organ, for that matter.
It is. It is lymph tissue in a way, I guess.
And it's full of cells that are doing stuff in there. Who would have thought?
I don't know. All right. Well, we're going to be speaking to Dr. Rahul Roychoudhuri at the University of Cambridge in just a moment. But before we get to that, aiming to take your chimeric antigen receptor T cells from bench to clinic? Get a free wall chart summarizing the processes involved in producing CAR T cells for therapy. To learn more, visit stemcell.com/CAR T cell-wall chart.
For the second part of our episode today, I'm really, really happy to be welcoming for this interview Dr. Rahul Roychoudhuri. He is professor of Cancer Immunology at the University of Cambridge, and I really like his work. He has excellent work on immunotherapy, on particularly transcription factors that I found very fascinating. But we're going to be talking today about cancer therapy and related topics. Thank you so much, Rahul, for joining us for the Immunology Podcast.
Thank you, Brenda. It's a real pleasure to be here.
Thank you for coming on. All right, Brenda, I know you're ready for immunotherapy.
Well, I mean, always. We aren't we always? Isn't like half our episodes, but let's get started a little bit with the basics. So we all know, I think our listeners are all very aware that the immune system is our best tool against cancer. And we have made such enormous advances on cancer immunotherapy in the last few decades, few years, and new things come all over. All the time we have new advances. So maybe do you want to start sharing with us what are the interests of your lab and your research around cancer immunotherapy that most excite you and that like you really feel are going to push the field forward?
Absolutely. So I agree with you, Brenda, that the advent of checkpoint blockade immunotherapy just created an explosion of interest in immunotherapy as its scope has gradually extended to a greater and greater number of cancer histotypes. But we're now sort of seeing a plateauing off of that explosion, right? With some patients with some cancer types, especially those with hot tumors that are neoantigen high, being immunotherapy sensitive. And these guys do really well on checkpoint blockade immunotherapy, for example, with anti-CTLA4 or anti-PD-1 therapies. But who are being left behind at the moment are those with typically insensitive cancer types, cold tumors, neoantigen sparse cancers. And one of the interests, I guess, the sort of general interest of the projects in our lab now aim at understanding how we could leverage immunity to benefit those patients. And so we're entering this synthetic immunology approach.
So, do you think it's a matter of not having the right checkpoint, or that it's a different problem after this initial burst?
Right, that's a wonderful question. Yeah, so I think the scope of checkpoint inhibitor therapy, the extent of that scope has not yet been reached. So in part, our lab is very interested in understanding additional modes of immune suppression within tumors. But I think that approach still applies to those with neoantigen moderately dense to dense hot tumors, or at least neoantigen hot tumors. What might be more important for those with neoantigen sparse tumors is resorting to alternative approaches which aim to create novel immune reactivities, either through neoantigen vaccination, where we know there are, neoantigen sparse tumors nevertheless likely have passenger neoantigens that can be targeted. And so we're really excited about the potential for vaccination. We're also interested in cell therapy, and a number of projects aim to endow T cells with synthetic effector functions, particularly suited for cancer, or to endow them with enhanced persistence and the ability to appropriately migrate within tumors. And finally, we've been asking whether we can extend immunotherapy scope from advanced cancer patients with existing metastatic disease to preventing that metastasis from ever occurring in the majority of patients who have early cancer, which is diagnosed and treated with curative intent, but who are at risk of getting metastases. So adjuvant therapy with anti-metastatic prophylactics.
So maybe we can take an example from your lab to illustrate this idea of making cells better at their work, improving their persistence, or understanding some of the networks that will guide this persistence. Or so one of the works I'm familiar from your lab, because I'm close to the Treg field, is your work on BAH2 as a transcription factor that in the case of Tregs is very important for their function, but as many transcription factors do, BAH2 has multiple other has other roles in different circumstances. And I think this as an example of, for example, a transcription factor that might also help a persistence or generation of memory in conventional cells. So maybe would you like to use this as an example of maybe you can target BAC2, maybe you can use this also as a synthetic biology approach. How can we model, or how can we influence the cells from their functioning to make them better tumor killers in a way, or better therapies?
Oh, well, you touch upon a challenge we've been trying to crack for maybe a decade now. So as you referred to, a while back, we showed that memory T cell responses require this transcription factor BAC2. BAC2 appears to act as a quiescence factor in T cells. So what it does is, in a subset of cells which express it, so it's dynamically regulated during T cell differentiation, it imposes a quiescent state, not a very functional state. And we now realize through analysis primarily of CD8 T cell responses, initially to acute infections in mice, but now also chronic responses to viral infection and cancer, that the maintenance of T cell responses requires this division of labor between quiescent cells, which remain relatively non-functional, and their more differentiated, functionally active progeny. So this division of labor enables those quiescent non-functional cells to maintain their genomic integrity, maintain a metabolism suitable for a long life, and they remain potentially within lymph nodes where they can last for a very long time. They self-renew, in other words, give rise to a cell of the same phenotype, while also giving rise to more differentiated progeny, which migrate to tissues, extravasate blood vessels, and then kill target cells. And we found that BAC2 was very involved in maintaining that quiescent cells, that quiescent state of those memory cells. So a while back, we asked whether overexpression of BAC2, this memory factor, would help a CAR T cell response, really, I should say, an OT1 T cell response against chicken egg ovalbumin expressing tumors. And what we found was that the cells overexpressing BAC2, in fact, could not differentiate effectors. They were locked in this quiescent state. And so for a very long time, we've been trying to work out whether we can utilize this quiescence factor function of BAC2 to make a long-lived response, but which is nevertheless also functional. And that's got us into this whole territory of autoregulatory circuits and dosing BAC2 at the right levels. And we think we've just about cracked it, and are hoping to write up a manuscript with some of this data.
So then it sounds like you have to deliver BAC2 though for it to work, right? Then so
you have to deliver BAC2, and you have to deliver it using some form of transgenic approach in cells for cell therapy.
So you're thinking like a gene therapy type of thing here, or like...
Yeah. So exploiting this fact that CAR T cell therapies require you to engineer the cell with a chimeric antigen receptor, you could also deliver other payloads, genetic payloads within the cell that, instead of redirecting their target specificity, also change their cellular differentiation state or function. So this is really, we like to think of it as cell therapy version 2.0, because really we're stepping beyond the sort of target challenge to this brave new space we and other labs and even industrial entities have entered, which is of synthetic cell therapy.
I have to say, this is a clear example of the trade-off, right, between effector function and longevity. I think this is usually it sounds logical when you think about it, but these are the mechanisms by which the dichotomy is maintained, right, by transcription factors like this and other associated ones that are really getting the cells from one state to another. So I don't know, it's a very clear example. I really like that when you have something so so clear,
Yeah.
so
So to
useful.
engineer the long-lived T cell response, we had to take a leaf out of nature and ask, how might you regulate and deliver this synthetic payload in a way that could enable more memory cells, but for those cells to lose that payload and then differentiate into effectors all during a response.
And do you have other research lines around this idea of maybe CAR T cells, or what other? I know that you've also been collaborating with groups that are doing metabolic interventions, or cytokines that work at under specific circumstances. So, what is kind of your current interest on other ways, other avenues to improve these cells using synthetic biology?
Right. So normal T cells, natural T cells, probably evolved to combat the threat posed by infection, and in particular cytotoxic T cells, intracellular viral or bacterial infection. And so they've developed approaches, cytotoxic functions, the ability to secrete certain cytokines. But not all of these functions are best suited to tackling cancer. And there could be more tailored approaches were evolutionary selective pressure at that time driven around the cancer problem, which it probably wasn't. I mean, we get cancer at a post-reproductive age. And so evolution probably hasn't had a great impact on shaping T cell functions around restricting cancer. And so, a couple of projects in the lab aim to create these synthetic functions of T cells, so we can have inducible promoters driving the induction of synthetic proteins, peptides, which have directly tumoricidal activity. Some of this is pretty early work, and we are trying to file a patent on some of those potential inventions. And so we are very interested in the idea of endowing T cells with synthetic effector functions that are tumoricidal, beyond their natural functions, killing cells, secreting normal cytokines.
Are you familiar at all with the DREAD receptors that have come before, those synthetic GPCRs that then couple appropriately downstream, but have, activation, ligands that are only for those novel receptors that you can't find?
Yeah, I mean, it's a very exciting, brave new synthetic space we're entering now. And cell therapy has remarkably been the venue where we can really get very innovative with gene therapy, because we have this opportunity to culture cells ex vivo, genetically engineer them. At present, the approved cell therapies all rely on lentiviral delivery of relatively small payloads, but with advances in CRISPR and CRISPR integration technologies, I think we'll be able to engineer cells with very large genetic payloads containing potentially modular functional payloads, many payloads at the same time.
So continuing with I change a little bit gears, but thinking about immunotherapy and what is the next step on the therapies that we actually administer to patients. I think one of the trends that we're seeing is that, especially for checkpoint inhibition, patients are being treated at earlier stages. I think now that the safety profiles of these therapies are kind of well characterized, and sometimes the benefits are so clear that patients are being they don't wait until it's completely out of control, so to say.
Yeah.
So, and I think it's important because then you have the differences between your primary tumor, your metastases, and we know that there's differences in the type of cells that compose each of these. What do you think is the along these lines? What are the next steps in our understanding of immunotherapy, checkpoint therapy at earlier stages?
Sure, exactly. So you referred to it as the next steps, but in a way, it's like a step backwards, because the focus of immunotherapy development has thus far been on tackling widespread metastatic disease in patients with advanced cancer. But when I'm forced to sort of think about the relationship between a cancer cell that's embedded in a large cancer mass, the relationship between that cancer cell and the immune system, you can imagine that cancer cell embedded within this large tumor is actually protected from immune attack by a vast immunosuppressive microenvironment full of inclement metabolites, a scarcity of nutrients essential for T cell growth and activation even, and immunosuppressive cell types like regulatory T cells and myeloid cells. But when a cancer cell metastasizes out of that primary mass of tumor cells into a new tissue, it travels via blood or lymphatics, it is actually divorced of that protective microenvironment. Suddenly it's in a very vulnerable state, right? It's in an immunologically primed host in a tissue in which it is not surrounded by an immunosuppressive environment. So it's suddenly very vulnerable to immune attack. And so there is, of course, great potential for using existing checkpoint immunotherapies at an earlier stage in the advanced cancer context, indeed, as clinicians call it, at the adjuvant, in the adjuvant setting. So after someone has had an early cancer, which has had limited spread, excised, and the surgeon there has gone, well, I have, to the best of my knowledge, excised every cancer cell in this person's body, but we know there are potentially metastases, micrometastases that have already spread earlier during cancer development. And those are either latent micromets and will take months to years to grow out, or they're just waiting to grow. Those cancer cells don't enjoy that immunosuppressive environment and are potentially very sensitive to checkpoint immunotherapy. But we also know from, for example, a lot of mouse models, that those micrometastases, the process of metastasis and the growth of new metastases, are sensitive to a distinct mode of immune assault to the cancer cells embedded in large established masses. For example, we know that T cells play this extremely important role in constraining the growth of large established cancer deposits. And while NK cells have probably a less significant role in that context, we know from mouse models in a beautiful paper published last year from Scott Lowy's lab that NK cells play an outsized role in restricting metastasis. So in addition to just using the same immunotherapies we've developed for advanced cancer patients in the adjuvant context, I think it's really important we start to understand what differs in our mode of effective immunity to metastasis versus established tumors, and to exploit those differences in the adjuvant context to really prevent this devastating thing from happening to patients, metastasis.
Well, that's what I want to answer more about, right? You talk about this one cancer cell inside of a big tumor that's a suppressive, hard to kill, right? And any given cell is protected by all its buddies, which is right. But a metastasis, like a metastatic cell or two, it's just floating out into your bloodstream without any of its friends around, in this model. So
very brave journey.
Why does the immune system have such a hard time killing all of those? It can kill every, bacteria in your bloodstream and make you, you get 10 to the third bacteria injected in your blood with a IV needle or something, right? Very small amounts, and no one goes and gets septic. Yeah. But these foreign cancer cells, which should read as non-host, they seed everywhere all the time. So to get to your point, there seems to be something different. And have you guys been able to start understanding that? It sounds like NK cells are part of the police, but what's happening with their patrols here?
Well, Jason, so I'm not sure that it's completely ineffective. It may be the case that actually anti-metastatic immunity is very efficient and picks off the majority of metastases. We don't really know the answer to that question. But we also know, for example, that when metastases establish themselves, having extravasated into a new tissue, they often sit there for months or years. not really growing very much. And it's hard to understand from a cancer biology perspective why that is. This cell has come from a large and rapidly expanding primary tumor mass. It escapes, it makes this brave journey across blood and lymphatics to the next site, and suddenly it sits there for months to years. We don't really know why, but I've always wondered whether the immune system has some role to play in that, whether there's a new round of immunoediting and a vigorous sort of war of attrition with a heightened immune response absent the microenvironment. So we're really interested in understanding sort of the ways immunity to cancer metastasis might differ. There are some really interesting literature about some fairly low bar approaches one can take to preventing cancer metastasis. We've been really inspired by some epidemiologic meta-analyses of the efficacy of aspirin. So, aspirin has been trialed in cardiovascular medicine, of course. Large placebo-controlled randomized studies have established that aspirin is an important prophylactic medicine in heart attacks, strokes, and it's widely used to prevent clotting. But one thing that cardiovascular medics do very well is this kind of focus on preventative medicine, which cancer biologists, because of the heterogeneity of cancer, the perceived heterogeneity of the pathogenetic causes of cancer, we haven't really got into preventive medicine in the same way. But the results of some of these cardiovascular protection studies have been quite interesting. And that is, for example, that in those who were on aspirin in these studies, if they were unfortunate enough to get a primary cancer diagnosis, and thousands of patients at that kind of age when they were taking aspirin did, their chances of distant metastasis were substantially reduced, 30 to 45% reduced if they were on once daily aspirin. And it's a bit of a mystery how that exactly works. There's well-established roles of platelets during the vascular phase of blood cancer metastasis. But we've been interested in whether aspirin might have an immunomodulatory effect.
That's very interesting. Kind of unexpected, I guess. It's nice to see that you have such a large population to draw this conclusion from because of the other studies.
Yes, yeah, absolutely. I mean, and another interesting study, again, from the cardiovascular trials, were research into IL-1 antagonism. So IL-1 was being trialed for a variety of cardiovascular indications, but while the study didn't meet its endpoint in terms of what they were looking for, what they did notice was a pronounced reduction in primary lung cancer carcinogenesis. And so this plays into sort of the role of inflammation in carcinogenesis. And so while cancer is a genetically heterogeneous disease, inflammation plays a fairly important role as a common denominator driving certainly a subset of cancers. And that can be intervened in, even at a population level. So I think there's scope also there, but that would probably be innate immunity.
I think also with colon cancer, they've seen this, right?
Oh, yeah. So aspirin has, so while the anti-metastatic effect has been detected in as a post hoc meta-analyses of RCTs of aspirin for cardiovascular indication, this ability of aspirin to prevent primary colorectal cancer in high-risk individuals, for example, with Lynch syndrome, has been tested prospectively, and it has been found to reduce risk of cancer by fairly large ratios, 40 to 60%.
Oh, wow. That's a lot.
That is remarkable.
Should we all be taking aspirin then?
I don't know. It makes a compelling case.
Considering having less of it from the bleeding risk that people are getting.
Right. So that's the downside of aspirin.
Right.
But when people have analyzed where the bleeding risk comes from, it's certainly the very oldest patients who have this sort of major bleeding risk from cancer. And actually, it's a relatively safe drug at low doses.
That's very interesting. So coming back to what you mentioned about adjuvant therapy and the implications for early cancer treatment before widespread metastases, I see there's many clinical trials looking into neoadjuvant modality in which you then are administering the immunotherapy before resecting the main tumor, and that actually has a very positive effect because then you have material from the tumor that can seed, can provide antigen for these newly activated T cells to recognize. So how do you see, in your opinion, how do you see this approach working?
Yeah, I think it's a very interesting point that there is more substrate for priming and especially de novo immunity if that needs to happen and if the immunotherapy is relevant for that to happen. For example, our paradigm for how CTLA-4 works is that it does help to recruit novel immune reactivities into the anti-tumor response operating within the lymph node, whereas perhaps anti-PD-1 works on existing effector responses that are PD-1 positive and unleashes their function in the context of PD-L1 expressing tissues and tumors. So it might be that different immunotherapies have different relationships with the presence or absence of tumor. It could be that in some cases, tumor burden, the scale of tumor burden is a problem for the immune system. For example, in cell therapy, which is more effective when there's less, not greater tumor burden.
So I think we're getting to the point where we could talk for 4 more hours or have to wrap it up. So that being said, we always try to end the conversation with some type of, get to know the scientist question or something else behind the scenes. And so I always find this to be the most interesting one. If you were not a scientist, what would you be?
All right. Very interesting question. I mean, when I was a toddler, I wanted to be a builder. And I certainly enjoy doing woodwork and DIY at home even now. And I joke sometimes with my good buddy, Tim Halim, who has a group at the CRUK Cambridge Institute, that if all else fails, if we don't get our next grant cycle, we can start a little company making garden rooms in all those gardens in Cambridge. So that remains a sort of potential alternative career option for me. But what I like, I guess, about woodwork and DIY tasks in general is they have a beginning, middle, and end, a defined end point to the work. Whereas, science can often feel open-ended.
Yes.
But what I tried to do towards the tail end of my postdoc, and what I encourage people in the lab to do, is think about each experiment as a defined task with a beginning, middle, and end. And really, whether or not the result worked out in the favor of your hypothesis, if you got results from that experiment where the negative control worked and the positive control worked, and maybe you threw in a special side experiment in it, which had an interesting question you were really itching to know the answer to, then maybe that should be viewed as a unit of success. And so, really science, it can feel very open-ended, but it is composed of discrete chunks. And we should really go home after finishing an experiment and analyzing it with the knowledge of a job well done.
That is such a great. I think this also answers another question we like to ask is whether you have an advice for our listeners. And I think this is a really nice way of looking at. Definitely the open-ended nature of our work does get to me sometimes because it feels like the work it just piles up. It's never there's no end in sight. And I also do enjoy manual task of defined projects. that, give you that kind of satisfaction.
And I think we should have the same pride of satisfaction in a negative result, right? I mean, knowing what things are not is equally valid, if not sometimes more valid, than knowing what things are.
Yeah. I really wish we could publish more negative data with the same glory that positive data gets. Although I will say, having jumped the academic fence, even when you have defined projects, and you think you're done, there's just another one right there the day after.
At the frayed ends of our knowledge are just yet more questions.
I mean, if this is what we signed up for to some extent, right?
We asked for this.
But it's never ending. There's always something to do. At least you're never bored.
Yeah. That's good. If we were building garden rooms, we may have built the very last garden room and saturated all gardens in this place. So at least with science, there's really no end to the work.
That is true.
Absolutely. All right. So thank you so much for joining us for this conversation today. So we had on board Rahul Roychoudhuri from University of Cambridge in the show. Very nice conversation. Thank you so much for joining us.
Not at all. Great to chat.
Thank you so much for being here. That brings us to the end of our show. Don't forget to subscribe to our newsletter, www.immunologypodcast.com, to get the show notes, including an episode summary and a link to all of the interview and Roundup Papers. You can also reach out to us on X at @immunopodcast or via email, info@immunologypodcast.com, with feedback or to just suggest guests. See you next time.